Thank you very much. And now it's time for something completely different, because the last two talks we're doing prediction of DNA methylation, and now we're going to totally switch gears and do prediction of transcription factor binding. So, hold on to your seatbelts. No competing interests for this talk. All right, so here's my lab's view of what transcription is, our little segment of gene regulation that we're interested in. You've got a transcription factor that binds DNA at a transcription factor binding site. The transcription factor recruits RNA polymerase II, and the RNA polymerase II produces RNA, right? And I hear that there are lots of exciting things that happen after that point, but that is somebody else's problem. That is stuff that happens in other tracks here at ISMB, and frankly, we have enough to chew off just trying to understand and predict everything that happens before here. So I'm going to talk briefly about ChIP-seq. So I know ChIP-seq was mentioned in the talks earlier. How many here have worked with ChIP-seq data? Oh my goodness, almost everyone. Okay, so the very short version of this is that we can figure out where proteins of interest in a population of cells are near certain DNA sequences by using the sequencer essentially as a way of counting that. And we get a nice little map at the end of it. And this is the most exciting type of data for me, because this is the kind of data that will let you understand what is it that is driving that key process that we saw in the previous slide, and what you can maybe change to make things change in the output. All right, so ChIP-seq is very exciting. So are similar techniques like cut and run. I'm at a cancer center. One of the nice things about being at a cancer center is it's very easy to get samples, right? When you're working on certain sorts of diseases, or you want normal tissue, it's kind of hard to convince people to, give up a little chunk of their normal liver or normal bone marrow or whatever. But, when you're working on cancer, and they're like, yeah, we think you have a little cancer there, and we want to do a biopsy and take it away. They're like, yes, take that out of me. You can have the sample. I don't want it anymore. But still, even with ready access to tissue, we don't always have enough cells to use for doing things like ChIP-seq, and enough to understand what is driving changes in gene regulation. What we do often have, however, are enough cells to do other nice techniques like RNA-seq, techniques to look at open chromatin, right? And our goal in my lab for some time has been, can you use these sorts of techniques? Can you use data from RNA-seq and open chromatin data to predict a sort of gene regulation, things closer to gene regulation, like where transcription factors are present? And by the way, these numbers were determined using Cunningham's law. And for those of you who don't know what that is, I'll explain it. I had a hard time finding numbers for stuff like this in the literature. It's actually, people don't really like to be nailed down on exactly how many cells you need to do for something. And then I asked on Twitter, this was back before Twitter was taken over by the current regime. And I'm like, so anyone have any good references for this? Or, how many cells do I need for TF ChIP-seq? And people are like, you wouldn't give any straight answers. So I just made up the numbers for the first version of this slide, and I posted the slide to Twitter, and I'm like, hey folks, what do you think about these numbers? People were like, no, you're wrong. You need at least 1 million cells to do transcription factor ChIP-seq. And I did DNA-seq once with only 30 cells, and it worked great. So that's why we have such weird numbers here. If you disagree with these numbers, please email me. Anyway, one other thing to mention here is this is the number of cells you need per transcription factor, right? So if you want a full, if you want a full complement of 1,400 transcription factors, first you're going to have a problem because no one can do this for all 1,400 transcription factors anywhere. But even if you could, you have the problem that you would need, billions and hundreds of billions of cells, and it's not going to happen, even for hundreds of transcription factors. All right, so this is a problem. Many people here have probably heard of the futility conjecture that was originally proposed by Wyeth Wasserman about how there are lots and lots of motif matches in the genome. And most, because people have been doing transcription factor motif analysis for more than 40 years now. Actually, it started before I was born, so it's actually more than 45 years now. It's really easy to determine this based on my own age, unfortunately. And it doesn't work so well in some ways because there are lots and lots of good motif matches that aren't actually biologically meaningful. So that was the futility conjecture. Looking into ENCODE data, we found something else, which we call the dual futility conjecture, which is not only are there a lot of motif matches that mean nothing, there's a lot of biochemical evidence of transcription factor presence all over the genome in places where you don't have good motif matches, right? So if you look over here, you can see for ChIP-seq, right, in ENCODE data, something like more than 70% of ChIP-seq peaks have a good motif match for ChIP-seq motif, and it's kind of downhill from there. And all the green ones here, right, those are ones where the ENCODE data, less than half of the peaks in ENCODE have a good match to the motif, right? And then you have all of these red things, and I get in trouble for calling these transcription factors because they are not. They're DNA binding proteins. We should be able to predict them just like other transcription factors, but they have no sequence motif. So if you want to predict those, obviously you can't do it based on sequence motif, right? So, about 15 years ago, people started making some amount of progress in being able to predict meaningful transcription factor binding sites. And they did this by adding epigenetic information. And the most important information was open chromatin data. So it's kind of this generation of tools, starting with the Boyle method here and Centipede. And this is a paper this is from the Hint paper where they show it. Show this, compare themselves against this whole generation of tools which rely on open chromatin footprinting. And the way it works is you scan across open chromatin data for some cell type, and you look for a jump in. Is this working now? Ooh, you look for a jump in open chromatin, and then a big drop down to 0 of open chromatin. Right, and the model is that transcription factor here is protecting the DNA from being chewed up by DNase or by the transposase that happens in a TAC-seq. Right, and then once you've identified these open chromatin footprints, you can compare that against a number of different motifs. This works really, really well for those motif matches where you have this open chromatin footprint, right? Which is not most of the motif matches. So most of the places where you find transcription factor binding in the genome, you don't have this nice signal. So it's great that they found something that works really well here, but we want something that works everywhere. So how do we move forward here? So there are two things we wanted to do to move forward, and these are mainly in terms of reconceiving the problem a bit. So before this work, a lot of times when people are trying to do prediction of where transcription factor binding was, they would conceive it in the way Bill Noble earlier talked about today, which is the chromosome by chromosome prediction approach, right? So they might say, okay, we're going to train on chromosomes 1 through 13 and predict on chromosomes 14 through 22, and here's our AUROC of 0.96, and isn't this great? Okay, that's a very unrealistic problem in the real world, right? The sequencing core is never going to call me up and be like, hey, Dr. Hoffman, we're sorry, we lost your data for chromosomes 14 through 22, and you're going to have to impute it, right? It's never going to happen, right? So I would argue it doesn't make a lot of sense to try to focus on that problem, right? What is a real world problem is one where you can collect this for some types of samples, like ones for which you have cell lines and get lots of cells, and then you have new samples, right, where that is impractical. But you can possibly get data for open chromatin and RNA-seq data. And there is actually an amount of data out there, number of samples or tissues out there for which you can find those two types of data, but not a third. The other thing we wanted to do is to start using global cellular state as measured by transcribed RNA, right? And sort of the intuition here is, at an extreme example, if you have some transcription factor and it's not expressed in some cell type, you should be able to create a machine learning system that will predict that there's no binding of that transcription factor in that cell type. Modulo a few questions about the steady state we're looking at things. And stuff that is upstream of that transcription factor. Hopefully you should be able to understand that as well, right? If something that is upstream of transcription factors has different global expression in some cell type, should probably change the way that transcription factor is bound, or something that cooperates or competes with it, or something like that. So I'll show you an example of how we try to incorporate this sort of knowledge into our system here. So here we have 10 different cell types. Right? Someone asked me earlier today what my favorite model organism is, and I said human, because as a computational epigenomics person, you can just download more data for human than anything else. I mean, it's great that mouse is very tractable for experiments relative to human, right? But like, I can download 3 times as many ChIP-seq data sets for human than mouse, much better. So here we actually have 10 cell types, which I don't think you could get for any other species. And we have 5,000 genes. So we just, to make the problem easier, focused on the 5,000 genes with the most variance over these 10 cell types, right? And then now we're looking at 4 different segments of chromosome 5, just to show you what this looks like in different places. And we have the same 10 different cell types, and we have signal of ChIP-seq in 100 base pair bends, right? And what we want to learn is there a relationship between the pattern of presence of the transcription factor, here is MYC we're looking at, across those 10 cell types, and these 5,000 genes, right? And essentially what we do is quite simple. We do Pearson correlation across the 10 genes for each ben and for each 100 base pair ben, and we get something that we call the association matrix here, just to make life easier in the way we talk about it, right? And so anything where there's a p-value greater than 0.1, we treat as NA. Everything else, we have either positive correlations or negative correlations between the gene being expressed and the transcription factor being present in that position. So that's training this aspect. The prediction is we add an 11th cell type for which we have the RNA-seq data, but we don't have the ChIP-seq data. How do we make predictions there? And I'm going to show you an example of how we would do this in two different 100 base pair bins. Essentially, we look at which are the non-NA genes in the association matrix here. You can see some with a positive correlation between the gene and ChIP-seq signal for MYC at that 100 base pair ben, and some with negatives, right? And then we compare that against the rank of expression in that cell type of these particular, in this case, it's like 140 genes or so, right? And if there were perfect correlation between those two things, it would look like this, right? That's not what we see. In actuality, you see something that is a lot more like this. This is the real data for this ben, a lot noisier, but you can still see that there is clearly some direction to the correlation, right? So that's GM12878 at this position, and we call that Spearman correlation here. We call it expression score, just to make it easier to remember where it goes into the rest of the model. Right here, we do the same thing for genes in K562, and then if you look at the held out data, you can see at this bin, you have binding in GM12878, but you don't have it in K562. So this works really, really well for this one cherry-picked region that I've picked out of all 100 base pair bins in the genome. Does it work on its own? No. But we combine it with lots of other features, and we combine everything into a neural network. So we have the expression score, we have information about chromatin accessibility, we have information from genomic conservation, We even include the motif scores that I was bashing a few minutes ago, and this other thing, the number of cell types with transcription factor binding at that position. So this is what I was talking about, reconceiving the problem. You can add in a reference set that you can look at and see how often is the transcription factor in question present here in other cell types in the reference set. All right, so this was the first version of virtual ChIP-seq, and we combined all of these features with the world's stupidest neural network, which is simply a multi-layer perceptron that is fully connected, and we did searching over a variety of different hyperparameters. It's in the paper if you want to look at it later. You don't need to take photos, but you can if you want. And I'm just going to talk briefly here about assessing performance. All right, so you've seen here a number of people have talked about assessing the performance of their methods today with precision recall, and I'm just going to talk about why that is so important for this problem in particular, and it's often important for genomics and biomedical prediction problems. So I'm just going to give you an example since people talked a little about PR earlier, is 3 of our models here. We have EZH2 with AUROC of 0.998, CTCF 0.982, whatever. If you compare these things in the precision recall space instead, you will find EZH2, something that has an AUROC that rounds to 1.00. Actually, in the precision recall space, it is worse than a random predictor, right? So something has clearly gone wrong here. And essentially what has gone wrong is that when you're using RC analysis, there's a true negative in the denominator, the false positive rate. And if you're looking for a needle in a haystack, like you're looking for EZH2 binding that occurs 200 times in the genome, you are going to very, very quickly get to a very, very small false positive rate. And this is a problem that precision recall analysis doesn't have. So the input to our classifier looks kind of like this. You can reconstruct things as a table. We have, for each, so we have 200 base pair overlapping windows now. For each window, we have multiple cell types that we're training on, some columns that are specific to the region, so things that are based on sequence or things that are based on our reference panel of ChIP-seq data, and then a set of things that are specific to the cell type and the region. So that's stuff like the expression score, stuff like the attack-seq signal on that region. And we did some analyses to look at ablation study to look at which features are most important, right? And here is CTCF, which I showed you earlier, looking at how AUPR decreases as you get rid of different features. So if you get rid of ChIP-seq and the expression score, and by ChIP-seq, I mean ChIP-seq data from the reference panel, things perform a lot worse. And some of the other things that we looked at were not quite as important. Now, you should never believe anything anyone tells you about a transcription factor prediction method that where they focus on CTCF, because it's fundamentally different than all the other DNA binding proteins, so remember that. But this is actually a trend that we see in lots of different transcription factors for our model. And in fact, you can summarize it this way by looking at whether there's an increase or decrease in performance across multiple models from excluding these things. The expression score and the ChIP-seq reference panel are really important for improving performance here. And then some of the other things like integrative methods, like including the output of hint or including cream in here, can make performance worse. And that's not because there are inherent problems with those methods, but I think it's because the information that those are providing are already provided to our model by the incorporation of open chromatin data directly. But it does make things simpler to not look at those data when we reran all the models. So we did do that, and we because we're inhuman, it was nice enough for us to have several cell types that we could hold out from our prediction. And this is sort of the output at the very end, is we have a set of DNA binding proteins for which we have AUPR of greater than 0.5, right? So that includes things like CTCF, includes things like MAX. It also includes things that like SMC3 and RAD21, which are not not transcription factors. They don't have a sequence motif, right? But it turns out from the data from other cell types, they're very strongly aligned with CTCF, and you can do a really good job of predicting them, right? Here's a set of models. So most of the transcription factors we looked at, we have AUPR of 0.2 to 0.5, which I would characterize as promising, not great yet, but promising. But still, if you look at what people, when I talk to biologist colleagues of mine, and they're like, we have this interesting region of the genome, we want to know what transcription factors are driving it. Often what they end up doing is just a sequence motif analysis, right? And if I were going to bet on the results of experiments, I would be far happier to bet on something that relies on something like this than looking at sequence motifs without any other information. All right, and then there are set for which the prediction is really kind of poor. That includes EZH2, our second worst model, and remember this is the one that had AURRC of 1.0. So don't use AURRC for this. All right, so for the 36 DNA binding proteins for which we had the best prediction, we had Matthew's correlation coefficient of more than 0.3. We went further with those. We did some analysis I'm going to show you here. First is something we're interested in is, is some aspect of the data or the model chiefly responsible for the performance of the predictions here? And we decided to look at correct predictions where we lack any one of these particular features, right? And we found that there were places where we made correct predictions, where there wasn't TF binding, where there wasn't chromatin accessibility, where there wasn't genomic conservation, or there wasn't a sequence motif, including this whole set where there's 100% that lack the sequence motif because there is no sequence motif. But you can see that some of those other things vary by the transcription factors. So our model is able to learn something for MAPK that doesn't MAPK, excuse me, that doesn't rely on chromatin accessibility so much, and then there are other things where some things are more or less important. The other thing we wanted to do is make this accessible to the world. Everyone here I know has experienced problems installing software, bioinformatics software. We did try to make this easy for other people to install, but most importantly, we tried to create annotations that people could use without actually installing any software. So we did prediction on 33 roadmap epigenomics tissues where we could find the sorts of data that we needed, and we predicted 36 DNA binding proteins in these tissues. And if you go to virchip.hoffmanlab.org, there's a big button that you can press here, Virtual ChIP-seq Track Hub. You press it, it adds this nice track hub to the UCSC genome browser, and then you can turn on whatever DNA binding protein you're interested in. So if we say turn on NRF1, you can turn on predictions in this or that or the other. And so if you are one of the people who is intensely interested in fibroblast of skin of abdomen, like this is the track for you. You can turn that on and you can predict in fibroblast of skin of abdomen to your heart's content. Here's the results of something, where this look in this region, there is a prediction of transcription factor binding in fibroblast of skin of abdomen. And you can see this, matches up with a place where there is real evidence of transcription factor binding from ENCODE. All right, so what is next? So as mentioned, so the main goal of this was in terms of reconceiving the problem of predicting where transcription factors are present, and we used the stupidest possible model that might work. So the next step, obviously, is to start to use more complex models and see, how much other performance we can eke out of this problem. So, incorporate for position dependency, stop pretending things are IAD when they aren't. And we want to add lots of other nice other sorts of features that might have a role here. One of the most promising so far is DNA shape, so physicochemical properties of the DNA at particular locations, and we want to also make this work at much lower resolution. So at this point, I'm going to sum up and leave some time for questions. So using ChIP-seq data and modeling global gene expression instead of focusing on sequence preference lets us do transcription factor binding prediction more accurately. And we have produced a annotation set that is available on our website for 34 roadmap tissues for 36 different DNA binding proteins. So I'll hope you check it out. I want to thank Mehran Karimzadeh, who was formerly a PhD student in my lab, who did this work. And finally, I want to thank all of you for your kind attention. Thank you very much.
Great talk. Thanks for the exciting method. Yeah, you can leave it here on this plot. You said that with EZH2 transcription factor, you get an APR of 0.003, and that's bad. But I think it depends on the ratio of the positive for that specific protein. And also the comparison between the different transcription factor is problematic because the random baseline for APR is the ratio of positive in populations.
Yeah, so this is a question I would say about when people make a prediction, like what does it mean, like, and how when you're getting someone to rely on that prediction, like if someone is going to do a laboratory experiment based on your prediction, what should they expect? So yes, you can compare ROC between different different transcription factors or even different evaluation sets more easily, but I would argue that it's not often what people are interested. If you go to a biologist and say, let's say EZH2, like I have a method and it will predict where EZH2 is binding and it has AUROC of 0.99, right? And they do 100 experiments, they are going to be very disappointed because they're going to find that not 99% of those experiments did they get the correct answer. In fact, they get correct ansors something more like 3%.
I agree that the AROC metric is not appropriate metric.
Yeah.
It should be APR, but APR are not comparable.
Yeah. I agree they aren't comparable. But some of these, what it does, it reflects in some way that some of these are much more difficult problems than others, right? Like the things over here, we do poorly, but I think that they are inherently more difficult. And that's what we see. Thank you. Yeah. Yeah, I've been asked that a few times. We have not really focused on that. I mean, there's kind of, I think it might be interesting to try to do some sort of cross-species learning for something like this. I'm now inspired by the previous talks I've seen here, because people will ask me that before, and I'm like, well, if you don't have several, like if you're working on zebrafish, for example, because people ask me about zebrafish, and they're like, can we predict this? And I'm like, well, you need several cell types for which you have all the data to train on at least, right? And they don't have that. But maybe we could do some sort of prediction across species, and that would be helpful.
Like when I see the EZH2 and soft 2, I don't know, maybe the data set wasn't good that we trained it on.
Yeah, I mean, So, I mean, there are a variety of things you can do to look at the quality of the data. I don't think that that's, say, the problem with EZH2 and SOX2. So I think that there's a biological basis, or let's say that the things that are over on the left side, there's a correlation with, of these DNA binding proteins of the number of known factors influencing where they're binding of being relatively small, right? So you have things here like FOXA1, which is a known pioneer factor. When you have things over here like EZH2, which is only brought wherever it goes through a complex cooperation between lots of different transcription factors, right? I don't have any hope of really being able to overcome that with sort of this class of methods. And so I don't know. I can't give you the say ChIP-QC metrics for, even though we've looked at stuff like that for those particular data sets. I do think that it's related to difficulty of making this prediction.
Hi, Michael. I love this tool and the promise of this tool. So I was wondering, can you speak a little bit towards the generalizability of this in other cell types, specifically non-dividing ones like neurons? And then secondly, since transcription factors are dynamic, how do you envision this tool being used in terms of context, this specific or different cell states?
Okay. So the first question about different cell types, I mean, we didn't have, so essentially what I'm showing you here, I'm training on cell lines, and I'm showing you the performance on other cell lines, because essentially that's where I can get the test data, right? And essentially, this is a in distribution versus sort of out of distribution machine learning problem. The degree to which something is in the distribution of what I have tested these things on, is the degree to which I think these results will actually approximate what you have. So there are some, neural cell lines in here. So it's not, out of the question that it might perform well on real neurons, but it's hard to say. And I'm sorry, I can't remember the second question.
The second one was just about using this tool for different cell states within the same cell type.
Yeah, that's a good question too. I would think that I think it should be able to do well on something like that, especially if the cell type in question is within the distribution of cell types that we've trained and tested on, and that hopefully, like, the differences will mainly be in terms of the open chromatin data and the expression score, and hopefully that will affect the right things and you'll make a good prediction. But need more data.
One of my questions are already asked, so I'm just one question left, and it's basically, where are they located in the genome? Did you check that? Because some transcription factors always bind in the same location, while other ones are highly divergent. So especially promoter-associated transcription factors tend to bind in, in a promoter, yes or no, while enhancers are much more dynamically regulated. So did you look into these transcription factors, whether it could be that the ones that are performing well are more promoter-based, while the ones that are performing less are more enhancer-based, for example?
You know what? I don't think we actually did that and looked at where these are binding compared to gene annotations or gene models in a particular way. But yeah, I mean, just looking at these, I think you see kind of, there's some that are involved in genome organization that are, we do a very good job of predicting on, and other things that are like pioneer factors, and things that are like chromatin regulators, we perform more poorly on. So it's an interesting idea. We should probably take a look at enhancers versus promoters. Thanks.